Hello, everyone. In this lecture, we are going to talk about cell reproduction. And we're going to talk about cell reproduction in diploid cells. Remember those body cells that we talked a little bit about when we were talking about chromosomes. The two different processes that we're going to talk about today are binary fission, which is going to be in prokaryotic cells. The best example of prokaryotic cells are bacteria. And then we're going to talk about mitosis, and mitosis occurs in eukaryotic cells, which are going to be plant and animal cells. All right, so let's first talk about why cells need to divide. So in our human body, our cells are going to relatively be the same size. So let's say a human in comparison to an elephant. So does an elephant have bigger cells? No, they just have more cells. So our cells across are going to be typically the same size across different types of organisms. They need to stay limited in size because we have to be able to get oxygen into our cells and waste products out. We also need to be able to get food or energy into those cells as well. And so if the cells were so large, they would not be able to get that supply and demand that they would need to be successful and to be able to live. So if we kind of look at this, if we double the size of a cell, just say, for example, 1 millimeter to 2 millimeters, our surface area would increase 4 times and our volume would increase 8 times. So that means that we would need 8 times more nutrients and have 8 times more waste to diffuse in or out. And so it's really a time thing. We have to be able to stay small to be effective. So this is a really good visual when looking at why we need our cells to stay small. So as far as the surface area, you can kind of see in comparison. So the surface area of just one large cube, okay, is going to be about 5,400 microns in comparison to the surface area of 27 small cubes. The surface area is going to be 16,200 microns. And so you can really see the difference in between two cubes that have the same volume but have the different surface area. So think about this is represented by the little purple thing is a nucleus. So you can see how long it would take to get into the nucleus in comparison to this one. And so things are going to be able to diffuse in and out much more quickly when you have a smaller size. This is kind of just review from the last lecture, but we're really just looking at chromosome structure. Remember, the only time that we see chromosomes are going to be during mitosis or meiosis. So again, this is just going to show that succession from that stringy DNA, the chromatin. We're going to start wrapping DNA around those histones. The histones are going to start condensing together. We're going to see coiling and packing until we have that chromosome. Okay, so the chromatids come together, held together by that centromere. All right, so let's first talk about cell division in prokaryotic cells. So just the definition of division. So this is going to be that process by which cells reproduce themselves. Binary fission is the term that we give to prokaryotes dividing. So you can kind of see it's pretty simple process. So you have a prokaryotic cell, and then our DNA needs to replicate. Now, one thing you'll notice about a prokaryotic cell's DNA or chromosome is that it's going to be a circular chromosome in comparison to a eukaryotic chromosome, which has that X shape. So DNA is going to replicate because we still want to make sure that we have a complete copy of DNA in each cell once it divides. Then the cell is going to go through cytokinesis, and that literally means the splitting of the cell. And we have 2 identical daughter cells. So you'll hear that term daughter cells after we talk about cell division. So these are going to be genetically identical. You're also going to hear the term asexual reproduction, and that just means that they're genetically identical. and that the process of reproduction, two genetically different cells coming together, wasn't used, which is what happens in meiosis or reproduction in humans. All right, so now let's go into eukaryotic cells. Now, eukaryotic cells are going to be a little bit more complex. The first thing that we're going to talk about is the cell cycle. Now, I like to think of the cell cycle like the cell's life cycle. So it goes through different phases in your life, just like you did as an infant, a toddler, a teenager, into an adult or an elderly person. So this is really like the cell's life cycle. Now, the cell cycle or mitosis has two major periods, okay? So we have interphase and then mitosis. Mitosis is the actual process of the nucleus dividing. So then we're going to talk about the different phases within each major phase. So we're going to talk about the phases within interphase. So the first phase, or that we're going to talk about, are the G phases. So growth 1 and growth 2. So you can kind of see over here, we have our first gap, or our G1, and then you also have a second one. In these phases, the cell's going to grow. Metabolism is carried out. It's pretty much its normal process. Your chromatin is uncoiled. You're not going to be able to visualize or to see those chromosomes. It's just going to kind of look like mush, as you can see right here. During this S phase, the synthesis phase, that's actually when the chromosomes are going to double. So it's prepping itself to split. Because remember, when we split or when we create those two daughter cells, we want to make sure that this cell has all of the information that it needs, and then this cell has all it needs. So when it's in this phase before it splits, it has to double that DNA. A cell is going to spend most of its life in the interphase portion, okay? And this lasts about 28 to 48 hours. Some cells are going to divide much more quickly. So things like your skin have to reproduce much more quickly. And so their life cycle is actually going to be much shorter than, let's say, a neuron, okay? Here's a really good picture of a cell cycle. And so right here, you're going to see interphase, which is in blue. Then you're going to see mitosis and cytokinesis. And we'll talk about each of these in depth when we go into the mitotic phases. So again, during interphase, you have the growth, the S phase, which is when that DNA is going to replicate, and then your G2. Now you can see these little red marks right here. Those are going to be checkpoints. And so at these checkpoints, your cell's just making sure that everything is going okay. If it's not going okay, your cell actually won't continue into the cell cycle. It'll either edit whatever's going wrong, and if it's too detrimental, the cell will actually go through what we call apoptosis, which is cell suicide. It will actually kill itself so that it doesn't replicate a cell that might, let's say, have some sort of genetic should be genetically altered where it could cause certain genetic disorders or cancer. And so your cell's actually really good about going through these checkpoints. And if something's wrong, it won't go into that next phase. Now, obviously, sometimes our cells miss these points, and that's when we do develop diseases like cancer. All right, so let's talk about the first phase of mitosis. So after interphase, the first phase of mitosis is going to be prophase. Now, in each phase, I just want you really to know what it looks like visually, and that will help you. So DNA is going to be seen as sister chromatids. So you can see there right here, those chromatids are together. Our nuclear envelope is starting to break apart. Centrioles. So hopefully you guys remember this term from when we talked about the cells. The centrioles are responsible for pulling chromosomes apart during mitosis. They're going to be located right here. So you have your centrioles, and then coming out of those centrioles are going to be blue spindle fibers. And these fibers are going to attach to the chromosomes, specifically the centromere. So again, I just said that, but the spindle fibers are going to form between those centrioles. So you have your centrioles and then your spindle fibers. Now your cell spends about 25 minutes in this phase. So that is prophase. So what do we see? We can see the nucleus starting to dissolve. We see chromosomes now, so we know that that DNA is condensed. We're also seeing the presence of centrioles. So you can see the centriole, and then also spindle fibers are starting to form. Now we enter into the next phase, which is metaphase. So chromatids line up in the middle. So I always try to think of little tricks to help me remember what's going on. And so in this case, I think metaphase, middle, metaphase, middle. What's lined up in the middle? Chromosomes, okay? So you can see your centrioles are right here. You see your blue spindle fibers are going to be attached to the centromere or the chromosome, and it's going to be lined up in the middle. And mitosis, or excuse me, metaphase lasts about 10 minutes. These are averages, you guys, so don't really, take too much consideration of these. It's going to differ a little bit. The next phase is anaphase. So again, I like to think of little tricks. So anaphase away or anaphase apart, what's being pulled away or apart. The now chromatids, so you can see them, they're pulling apart. And what's happening here is I like to think of it like a tug of war. So these centrioles literally are pulling the spindle fibers, and as they're pulling those spindle fibers, they're pulling the chromosomes with them. It lasts about 13 minutes. This is an important phase because this is where you actually can get those trisomies or monosomies that I talked about when we were talking about chromosomes. And so that's where in that, more specifically in meiosis, but in this case too, you can actually have extra chromosomes or not enough chromosomes in your cells. All right, last phase of mitosis is telophase. Sometimes you'll hear telophase, but anyone's fine with me. So what happens now is it's basically the exact opposite of what's happening in prophase. So our nuclear envelope is starting to develop again. Our chromosomes are starting to kind of lose that distinct shape. They're going to be going back into that stringy DNA form. So I like to think what's happening in telophase, the exact opposite of what's happening in prophase. You can kind of see the centrioles, the spindle fibers are detached, and hi, Miss Mackenzie, she's bothering me. And so it's the exact opposite. So what are the phases of mitosis? We have prophase, metaphase, anaphase, and telophase. And I like to think of them as PMAT. So that's the order. So prophase, metaphase, anaphase, telophase, PMAT, because you will have to remember the order. And then the last phase, sometimes you'll see this clumped in with mitosis, but it's not necessarily or really part of mitosis, and that's cytokinesis. And that's when the cytoplasm splits. So cytokinesis refers to the splitting of the cytoplasm. Now, these are kind of important terms. So animal cells are going to use a cleavage furrow. So it's just an extension of the cytoskeleton, and it literally pinches in between the two cells. And then this is a little bit out of order, so sorry, this kind of should be at the end, but plant cells don't use a cleavage furrow. They use something called a cell plate. And what's cool about the cell plate is it actually will develop into the cell wall. So we know that plant cells have a cell wall, animal cells don't. And so this cell plate will actually turn into or develop into the cell wall. One other thing to note as well is that plant cells do not have centrioles. So at the end of cytokinesis, we now have two new daughter cells that are genetically identical that are diploid. So here's a picture of some cells going through mitosis. It's pretty cool. And that is the end of that. So if you guys have any questions about mitosis, let me know. I know it was kind of a lot, but as a review, you just need to know the two major types of cellular reproduction. So In prokaryotes, it's going to be binary fission. And then in eukaryotes, so plant and animal cells, it's going to be mitosis. What is that life cycle of a cell called? It's the cell cycle. The two major phases are going to be interphase and mitosis. What are the phases of mitosis? That's that PMAT. So prophase, metaphase, anaphase, telophase, and then at the end, it's cytokinesis. What is the product of mitosis? We have two daughter cells that are genetically identical that are diploid, meaning they have those two sets or a complete set of chromosomes. In a human, it would be 46 chromosomes. If you guys have any questions about mitosis, let me know.